Yeah, they're all here. Yeah. Okay, sorry, Bob, for that interruption.
No problem. I'll keep going rather than start again then.
Yeah, sure.
Okay. So speaking of the meningococcal conjugate vaccines, which were licensed in 1999 and which was the basis for my collaboration with NCMH, I then worked on many different conjugate vaccines, and I'm going to be talking about those here and talking more about important things in the relationship between structure in particular and immunogenicity, rather than hard and fast rules. I'm going to start with 3 maps in the next three slides. This is first a map in time, and it's a map of the vaccine development. And I think you can see that we're coming into the 2000s here. And the conjugate vaccines were licensed starting in 1999. So that in time, they're very recent, and they've been extremely successful. I'm going to tell you about the impact later. I'm going to move between some of these, and I want to mention that the Hib, the meningos, the pneumo, and also GBS, which hasn't been licensed, form the crux of all of the respiratory pathogens, which cause meningitis. And because of the success of these vaccines, there is now hope. And in October, the World Health Assembly approved a roadmap for really tackling and developing further eradication programs. But these really depend on the licensure of the GBH vaccine. In terms of deaths, the pneumo and GBS probably cause the most at the moment. But before I get into that, I want to just remind everyone, in the UK, we always start with the Jenner vaccine. But Jenner wasn't the only one. Just as with the coronavirus vaccines, there were many players and lots of funding. And but Jenner was successful. And I'll move now to a map of England, really the counties of England. And the 53rd parallel, which comes very close to NCMH, I believe, because I think Nottingham is right on the 53rd parallel. And these purple and red dots at the northern part of the country illustrate where the burials were over 90% from children under the age of 15 due to smallpox. And you can see that this north-south divide, as it was that marked by this area, is very prominent. There were some reasons for this. And one of the reasons had to do with behavior. The behavior in the South was that there were comprehensive vaccination programs when smallpox came in the late 1700s, a smallpox vaccine. Smallpox had been raging for hundreds of years. And another thing was that in the South, they continued to use the pest houses, which were evacuation houses from the time of plague, a couple hundred years before, 100 years before at least. And so this quarantining of people and self-isolation or mandatory isolation was effective. Now, the adults, because it had been around for so long, had herd immunity. The children didn't. And in the North, they became exposed because of different routes. And in terms of Nottingham, there was routes coming up here, which were turnpikes, and routes coming here and going across with all the waterways. There were navigable waterways. And this brought people into the area. And what happened was, it wasn't university students spreading it. It was actually young people, though, coming from London for work, who didn't have herd immunity because their parents were immune. And they spread that through the North. Another map, a global map this time, is of the cases, this was in 2017, of meningitis and neonatal sepsis. At the very bottom are some staggering numbers still, despite all the vaccines, that in that year, there were almost 500,000 deaths from meningitis or neonatal sepsis. And the UK is in an area which is now considered low. Same with Scandinavia, but this didn't happen by chance. Actually, Iceland, Norway, Denmark had big meningococcal programs that fed into the UK and also New Zealand. But there's been Cuba and Brazil in the Americas, and North America are also very susceptible, as was Northern Europe. And now you can see most of the cases are in sub-Saharan Africa. This is considered the meningitis belt, as well as Southeast Asia and the Pacific. So this is the real reason why we're doing this work, that I'm doing this work. And the conjugate vaccines have a common denominator, and that is that they all use capsule, whether it's from gram-negative or gram-positive organisms, which is the case here, showing GBS from cerebrospinal fluid left here, or from blood culture. And it's very interesting. This was of an 8-week female. That's not interesting. That's sad. Presenting with late onset meningitis, who was then given antibiotics. What's interesting, I wanted to say, is that the strep, which you think of as in chains, is in chains in the blood, but in the CSF, it was in singles or pairs, and I don't know the reason for that. Moving on, I have a few slides on the carbohydrates. At NIBS, we make standards. It's the National Institute for Biological Standards and Control. And we make, our highest level standard is a WHO international standard. These are ampuled in 1 to 5 mg per ampule. They're available for other labs to use, so that internationally the amount of Hib or any of these can be standardized around the world. And most vaccine-relevant polysaccharides are charged. They're anionic, most, not all the pneumos. And GBS and some of it aren't as negatively charged because it's spread. There's some neutral sugars in the repeating units. But all of these have single repeating units, except for WMY, which contains sialic acid, but also neutral sugar. They're very similar in structure. Most contain N-acetyls and also O-acetyls, and we're going to talk about that more later, Tony, and which are very important for the MEN-A and also the VI and 2 enterics. Well, they both have the same structurally similar or identical polysaccharide, which is the basis of the vaccine to prevent typhoid fever. We took these polysaccharides and ran Sekmal's viscometry on them under 3 buffered conditions: water, in which everything swelled up and became much larger. You can really see that pronounced in this way. Or 2 similarly ionic strength ones, PBS in the dark blue, and the heaps. The striking thing again is the water swelling up, but this one, the VI, is a huge problem. It behaves very poorly on matrices, and it's extremely viscous, although the measurements here don't necessarily show that. The fact that there are no bars here means that it didn't elute from the column under that condition. And again, in water, the viscosity increased at least 200, if not 1,000, a 20 to 200 times increase in viscosity, which makes a huge difference in manufacturing. And some of these you really don't want to take up to 5 mg per mil. So this shows the MEN-A, MEN-W, MEN-Y, and MEN-W. I'll just focus on the MEN-A, and I just really want to focus on the O-acetylated form, which the acetyl group is in red. It's in a prominent position. It sort of stabilizes that polysaccharide, which is in an extended form. This is from molecular dynamics simulation, and in that you put in a lot of water. You can put in divalent cations or monovalent cations to also give some stabilization. This contains a phosphorus group. And you also have to limit the size. I just want you to pay attention to 6 repeating units being limited by 60 angstrom box, so to speak. And these structures were done at the University of Cape Town. Compare that size with what you get in a crystal, where everything is very compact, not as many waters, and a whole 58 kDa protein with 3 domains only takes a space of 100 angstroms. But the carrier proteins so far have all been of bacterial origin, because after all, we want a T cell and mostly type 2 dependent response to convert the polysaccharide, which is independent of T cells and doesn't benefit from all the activation of that you get with the T cell dependent response. Another time, it'd be nice to talk about functional groups here, the active site, T cell epitopes. These things overlap. You want almost the conjugation sites, which might be a lysine or glutamic acid, aspartic acid, or occasionally a thiol, to be near a T cell epitope, I believe, but this really hasn't been worked out. Of the 39 lysines in CRIM, a cross-reactive mutant of diphtheria toxin, really only 6 to 7 are highly accessible and linked. One group that we've used a lot has been the tryptophan group, and intrinsic fluorescence especially, and a bit in near UVCD. I'm not going to be talking about that today. Tetanus toxoid, the tetanus toxin, is used in vaccines in its toxoid form. And so far, except for some small work done with an HC conjugate, the heavy chain C-terminal region, it's the whole tetanus toxoid that's used. It has 13 trips, but one nice thing is there's only one in the light chain, and the rest are in the HC region, which is nice because there's protective epitopes and T cell epitopes there as well. Now, the mechanism of immunity. It's pretty well established now that for these very beautifully and pure glycoconjugates, that the mechanism of immunity is through their engulfment or being brought into the cell by a B cell receptor into an activated dendritic cell, where they are processed in the various compartments and are presented, loaded onto an MHC2 molecule. Those peptides that are loaded are about 12 to 20 amino acids, with the additional amino acids for flanking regions. And what it is thought is that, and so that is then presented to the T cell, and other receptors help to stabilize that. This then produces cytokines and helps the plasma cells make antibodies. And here, if we just look at A and B, A shows just a protein peptide being presented, and B shows what I call a glycopeptide being presented. And the minimal epitope for the glyco part would be about 6 or 7 residues. I put together a list of what I think are the important topics in terms of relating to immunogenicity. And I want to answer some of these right away. Is protein conformation important? No, it really isn't important. And we have kind of moved away from that. It was something I thought was very important at the beginning. The folding is important in that it remains stable, that there aren't aggregation or particle formation. The minimal size of the saccharide protein epitopes I've just discussed. Does conjugation modify critical polysaccharide epitopes or create new ones? This is for the manufacturer to prove, and it's really important. What are the limits on the saccharide loading positions and stoichiometries? And optimization of these is very important. Can predictions be made on molecular size range effects on immunoprotection? And that's one of the things that I've been working on for decades, really, through sometimes troubleshooting investigations, but also an interest that we can take when we're not doing our sort of bread and butter work of doing control batch release work. And how does polysaccharide coverage of a protein affect its processing, intracellular processing, epitope accessibility, loading of the MHC, presentation and recognition, and all of these things lead to immunodominance or influence immunodominance. And these are really questions that are beginning to be answered one by one, but they will remain, and they're very important vaccines. And also, I think what you've been working on, Steve, and your colleagues, is how does the behavior and stability of glycoconjugates and the polysaccharide impact on their manufacturing and the quality of the vaccine? And also, perhaps through your Osberg work, an understanding of the optimal adjuvant interactions in stable forms. The protective immunity towards encapsulated bacteria, there are some expectations, like such as in between primary injection and the secondary, once the IgG kicks in for this booster dose, or even maturation, you would expect a 4-fold rise in titer in the IgG, not the IgM, because that wanes. You would expect there to be affinity maturation between the first and second dose, and especially in functionality, such as so that you would see enhanced opsonization coating of the bacteria, enhanced serum bactericidal killing of the bacteria with complement, and phagocytosis. You can get IgG isotype switching to IgG2 against bacterial polysaccharides, and also even in a polysaccharide or conjugate vaccine. And what's probably the most important thing is a demonstration of persistence and a boosting effect when it's encountered again for some. But for meningococcal bacteria, you don't have time to mount a response. You only have an hour in which your child mostly has to be seen to have their life protected. So they need antibodies that are circulating. And this is why we give in the UK a booster dose at 12 to 13 months. On the right here is something from a pneumo study done at Oxford. It's the kinetics of the IgG producing cells. These aren't IgG. It's IgG producing cells make in response to tetanus, diphtheria, and serotypes 4, 14, and 23F. And the peak of those cells is between 5 and 7 days. These are some of the assays which we do when I also put in sedimentation and other methods that you use to look at standard essential things. And in the conjugates, perhaps it's the most interesting from my point of view. And we use ELISAs to look at antipolysaccharide, anti-protein, now some multiplex in terms of the larger valency vaccines, and functional assays. And I'll present a little bit on cell-based methods and adjuvant adsorption, which some of your students have worked on, Steve. This is the mouse model that's used a lot. We would here use 2, manufacturers like to use 3, but this shows a couple important points. This is normally 0, 14, 28 days, and then bleed 2 weeks later. Between the first and the second dose, you see that greater than 4-fold rise here. It's greater than 20-fold. And not as much of a rise in the IgG here. I'm sorry, but the bactericidal bars didn't come in, because what you get is you get an increase, but what you get, the IgG isn't increased, you get much higher bactericidal activity, which is part of this maturation. This vaccine, which was made under the funding of the meningitis vaccine project for the meningitis belt, which has 26 countries. And 24 of those countries have had mass campaigns in the last 9 years, and 340 million people have been immunized. And for 6 years, there's been not a single case of MANA disease. So this is really fantastic. But the COVID situation is impacted in a very worrying way, although people are, and it's impacted here in the same way. The positive side is social distancing isn't spreading measles, meningitis, mumps, but the herd immunity, when people aren't getting vaccinated, is decreasing. And older people could be more at risk. Those who aren't being immunized could be more at risk. So here, there's been a reduction of about 5%, there's been about a few percent in DTP hexavalent vaccine, but 5% of MMR. And in Africa, there's been a 30 to 40% reduction. The teams from UNICEF haven't been able to roll out. And there's been a lot of studies, dozens of studies and clinical trials have been delayed. It's a huge worry for those programs. This is also the same vaccine. It just shows one of the critical quality characteristics, one of the most important for lab analyzing them and for the manufacturer to consistently make them. And it shows the increase in size. This is the MANA polysaccharide, activated tetanus toxoid carrier protein, a bulk conjugate, and the final fill. This is a beautiful example of how lyophilizing actually didn't create aggregates in this case. But it's so good in spotting problems with integrity or aggregation, insolubility or particle formation. Particle formation is one of the big defects. You have to keep your eye on. People giving it need to keep their eye on it. And so maintaining well-behaved molecules in good formulations is so critical. And this size can correlate with immunogenicity, but this is product specific. And you concluded, McCalman et al. Review concluded about everything should be really interpreted on a case-by-case basis, and I really couldn't agree more with that. This was our collaboration, which began with those first meningococcal conjugate vaccines with Connie Jumel in 1999, and we published in 2002. And it was the first publication of SecMols used for conjugate vaccines. These were the molecules, 2 Menc crims, one which is monovalent, 75,000 conjugated, so a monovalent, a monomer crim with oligosaccharides, which forms this sun type of, a so-called sun type of, in that case, unidirectional conjugate. And this one, in which the crim is more open and can oligomerize more readily, although it's not as pronounced as this. You see no RI signal here, but it's 350,000 here. So it's quite a few times that. Slightly longer oligosaccharides and bidirectional. And here is a MEN-CTT, which was made with deoacetylated polysaccharide. It's been very successful. And like all tetanus toxoids, if you use a standard, fairly standard column, you always get at least 2 peaks. This shows 1.6 million and 400,000. And these are referred to as lattice type, sort of cherry pie type configuration with multiple attachments. This just shows a small snapshot of what we do at NIBSC in terms of being the UK's national control laboratory, where we do independent testing, and all well-developed countries have such a laboratory. And our work is governed under a legislation, which has been national legislation, EU legislation. We're now under the Health and Social Care and still under the EU for a couple more months, well, 1.5 months. There's new statutory instruments, as they're called, being passed in the wake of Brexit, and we'll see what happens, but we don't know what's going to happen in terms of our relationship with Europe at the moment. where any European lab can test for another European lab. But from 1999, these were the vaccines that we released, the number of batches. Batches could range from about 20,000 to 250,000 doses. So if there's a problem with one of those large batches, it's very costly to the manufacturer to have to pull it out of production, especially if it was something found at the level of the bulk conjugate, which could have been used to formulate at least 4 batches, 1 million doses. You can see how the different vaccines evolved over time with the different colors. But of course, this also reflects what the UK have done. For a very long time, we were the only official medicine control laboratory testing this. That's a mark of the strength the UK has had in meningitis field. And really, it reflects the geography of the country and the epidemiology. This was one troubleshooting. Sometimes troubleshooting happens with routine analysis, and other times it might happen when something has happened in a clinical trial or the DH asks us to investigate something. This was from a clinical trial. It was 2 lots, it was 2 different lots of MenC vaccine that were used in babies, and lot 2 didn't give a protective response. Babies didn't get meningitis, but part of the clinical trial protocol was if they didn't meet the threshold, they needed to then redose them. And that is what has happened here. So we looked at this, but we weren't surprised. We knew that this vaccine wasn't the most consistently made, but it always passed the test. The column, the manufacturer wasn't resolving at all. Everything came out almost in the void volume. We used a better resolving column so that we could get something which we call the aggregate main peak, which you really can't see sadly down here. We then used SecMols and saw that it confirmed that one of these had a high molecular weight to monomer peak. And this really helped us understand. We called this a heptamer, and we found also there was dimer and monomer. Sorry about that figure. We then separated, this was a master's project, somebody based here. We separated them and looked at their immunogenicity of the high molecular weight fraction in yellow and low, as well as the formulated final fill or the bulk. And we found that this was the IgG, that the low molecular weight form always was giving a higher in the later stage in the boosting, and also had slightly higher SBA. This led us to want to look into a project, which was a PhD project, which was just published this year, in making a panel of conjugates where we looked at the effective chain length on immunogenicity, essentially. We wanted to look at the effective size, but in order to do that, we needed to purify different sizes of the chains. So this was for MenC. It's fairly labile. We could use 25 millimolar HCl, fairly mild conditions, and create sort of a whole panel of MenCs, which we also then had to confirm that they were acetylated enough. And so we have this smaller panel of polysaccharide chains to make conjugates with, because we wanted to have an ideal sort of panel. And when we ran all the detectors, Steve, we found something that was quite interesting. We published this, but I'd still like to get your opinion on it. We saw that the hydrodynamic radius, so this is for those, just go back, if you look at the conjugate size, it goes from 200,000 to 2 million. So we have 200,000 to 2 million in an exponential way for the hydrodynamic radius, or H.
Barbara, these dynamic light scattering radio.
Dynamic light scattering. These are with the SecMols.
But does your SecMols have a dynamic light scattering attachment to it?
No, we used the static light scattering. We used the 18 angle detector.
I know, but you've got that, but you have the additional DLS detector, which also online, like the viscosity detector.
Yes, viscosity detector.
But not the DLS detector.
No, we didn't have that. We only have a Malvern one, which is a standalone. Now we didn't have it at the time. And so what we've concluded, what was accepted by the referees, was that these smaller two are more spherical, that the radius by both measures was the same. But these were very different. And we took this to mean that these two were more asymmetric. We used a lot of the data that we got from those detectors. And I developed the stoichiometric model, whereby I used the chain length, the polysaccharide to protein ratios, the MWs of the whole conjugate, and through an iterative process, I let each one of those four conjugates find the best fit to different solutions. For example, A, which was the largest one, the 2.4 million one, fit best, and it fit very well to 3 polysaccharides connecting 5 TT molecules, this being a dimer and these being a monomer, but this being further back in space. And this polysaccharide, which I haven't done well, I think I've done better with the proteins because that's my expertise. And I don't know what to do with this in terms of making an accurate one, which I want to do, even if it's a sort of artistic thing. But each of these beads represents 50 saccharide units. What it does show is fairly close association, which you showed from your work with the meningos, where you saw when it was conjugated, it was more compact, which I was very happy to see that interpretation. And it shows these orange attachment sites. The seconds to the smallest one, the 400,000 one, fit okay to a 2 polysaccharide chains connecting 1 TT molecule. The other ones fit less well, but we included that in the supplementary to this. And what I would like to talk to you about is I want to get some data from the CRIM conjugates because I want to test this stoichiometric model further. And it just requires the data in terms of MW and hydrodynamic and RMS radius. And because I really want to put this, I want to test the model, but maybe put it into some software. And that would be nice if I could do that. Just see how useful it is as a tool, even if it helps people understand what they look like. The mouse model was used that I described to you, except it was a 2-dose mouse model. And so we used those 4 conjugates. And for this, which was made with a linker onto the protein, and then with carbodiamide coupling to the carboxyl group of sialic acid, we got an increase in IgG with increase in size. This was also seen in the overall with IgG, but this is one of those ones that is supposed to be particularly relevant for carbohydrate antigens. We concluded that in the space with molecules we were working with, that the larger size was more immunogenic. But this wasn't true with that MEN-CTT made with reductive amination before. Again, it sort of really has to be done on a product by product basis. And also, our size is actually well behaved. You know, we purified out any aggregates, but some of the ones that you see and we have seen are huge. And I would then question, I would believe that they would not be as immunogenic. And in fact, the reference vaccine was a commercial vaccine, and it was about 12 million, and it's not as immunogenic, but there were other features in common. This was stimulation of splenocytes that had been from those same mice, the spleens, and this was fresh. And then these lenocytes were pulsed without a mitogen, so there weren't a lot of proliferating cells, but with different size groups or that large reference vaccine or the individual components. And within each group, the larger one here gave a better response. This is larger than this. We concluded that this larger molecule also formed, was active at the level of the spleens, but not necessarily so in presenting. So using confocal microscopy, using the smallest and the largest, we looked at the localization of MenC on the surface of the cells. And to me, I don't want to make a lot of this. I don't think it's very quantitative, although some quantitative data was obtained. But it just shows that both in the large and the small, there was co-localization. My time's running short, so I'm going to speed up a little bit. We worked, as you know, with Robert Otto, who's a delightful person. And Steve, let me just move this, okay. He did his master's here as part of the Applied Biomolecular Technology course in 2004. And Saba, who I heard from yesterday, who's expecting her second baby and who's now writing rotation is for her PhD, was here in 2008. And what Robert didn't finish with adjuvant binding, Saba did finish. This was Robert's second paper, and it's actually a really well regarded paper. It's looked at a lot. I'm really proud of this work. We showed some possibly predictable based on electrostatics, but also important things using Hib and menC, CRIM and tetanus toxoid conjugates with both aluminium hydroxide and aluminium phosphate. It showed that phosphate binds a conjugate much more weakly than hydroxide, hydroxide being in the open symbols. At neutral pH, the anionic polysaccharide bound poorly to negatively charged aluminium phosphate, so no surprise there. And it did bind well to the hydroxide, which was positively charged. Carrier protein adsorption really wasn't based. It involved electrostatics to some extent, but other modes definitely. And tetanus toxoid bound much more strongly than CRIM. Binding strongly isn't always what a manufacturer wants. After all, you want something to be able to interact with the immune system. Phosphate could inhibit as little as 5 millimolar, could inhibit adjuvant binding. But if you're going to use an adjuvant, you got to demonstrate binding, and you have to demonstrate that there's a positive effect now in humans. Similar effects are expected for other vaccine-relevant anionic polysaccharide conjugates. Calvin, who was supervised by Judith Waite, was a fantastic student as well. And he looked at the stability of the meningococcal vaccine. And he initially characterized it as 2 million. It's a very, very well-behaved and extremely immunogenic. Those 240 million people have been protected, and they've only had one dose. We looked at the adsorption in saline after 2.5 hours or after overnight on a roller to increase the sort of surface area, or with trisaline, which was the formulation. The formulation was saline. PBS was being used for the immunogenicity in mice, which I was a bit worried about because of the effect of phosphate, which you can see it does prevent adsorption. And this was aluminium phosphate, so we wanted some absorption. We went to trisaline so that we had a saline base. All three had a saline base, and this acted very similarly to saline, although the pH was slightly higher. As you know, saline is a broad pH range. And so at the dose 2, which was the important one, there was a statistically significant difference between the PBS plus adjuvant and the saline plus adjuvant, so the green and the gray. And PBS gave a higher one, and actually I felt fine about that then, that we weren't not seeing something we should see. This vaccine is being formulated, has been formulated, and is being used with aluminium phosphate. But interestingly, the second generation vaccine, which is a pentavalent ACWY and X, which is in phase 3 clinical trials now, isn't relying on an adjuvant. And most of the ACWY vaccines, they do not use an adjuvant. Steve, I just wanted to let you know that we treat our students well here.
Yeah, but who's buying the beer? Is that Calvin buying? I hope he's buying you one, actually, Barbara. Anyway.
I was probably a driver.
Is that in the bar area? Do you ever, didn't know you had a bar at the NRBSC?
No, we have a bar, but we're not allowed to serve drinks here unless it's a special occasion. But this is a nearby one. Not near enough to walk, but yeah. Anyway, yeah.
Good old Calvin.
Yeah, I know. To sort of finish off with the impact of meningococcal conjugate vaccination, before I go on, just a few slides on the typhoid, if that's okay. I wanted to show you what happened in the UK after the Mense was introduced, which is the blue, which was the most deadly serogroup. And it does take a few years till the numbers drop. But then by 2008, there were no deaths in under 18s from serogroup C. And this really showed the possibilities for the first time. And then concurrently, I suppose, well, the MEN-A conjugate was introduced in 2010. And the epidemics there, I think I said, could be 2,000 to 20,000 to 100,000 every few years. So to be dropping down to no positive cultures from cerebrospinal fluid is really fantastic. Of course, then you have the other groups, but hopefully the ACWIX pentavalent will take care of these. However, the pneumo and this group B strep are still. So I really would like, I mean, I think it would be a dream. It won't happen in my time, but really to have a combined meningitis vaccine. And I don't see why it's not, why it couldn't become a reality in terms of interactions and immunodominance. I think that is a possibility. But is there the will? So I wanted to pause here, and maybe I'll take my slides down so I can see. Hang on.
Okay.
I want to stop sharing. Can you stop sharing for me?
I can. You can too. If you've got the square and the arrow at the top.
Okay.
If you just
Yes.
press that. Okay. Now we're all multi.
Okay. Yeah.
Multi-picture. So what's now, Barbara?
I just wondered if anybody had any questions for that part.
Any questions, folks? Tony reels out a hot sheet of 15 questions.
Yeah, I do have a couple of questions, but the main thing is something that I fed back to Barbara last week or so. Mainly on the biological side of this, all of the work that is here is essentially a structural approach. My question is, if we look at the biological side, how do the enzymes put these things together and take them apart? And could knowledge of that process, the biological enzymatic side, actually have a potential therapeutic effect? For example, if there was an esterase, if there was a specific esterase that took the O-acetyl groups off, that could potentially destroy infectivity and binding. And that would potentially be a tool that could be looked at perhaps, to combat disease.
Mm. I see what you mean. The manufacturers will be thinking about the transferases.
Right.
Okay.
Yep.
But not about the esterases. And I think this is something important that should be considered either during fermentation and, as you say, it's a possible means of control. So I can't add to that because we look at what is in the vial pretty much.
No, absolutely. I think that's right. It's when I tried to look up what there is, and it's essentially a blank. I think there's certainly for COVID, there's practically nothing. And because there are analogies, I think it may be valuable to squeeze that a bit harder, if you like.
Mm-hmm. Right. Okay. I will ask the manufacturers we collaborate with about this, and also those who have published recently about this. I think it's great that you brought it up. Thanks for that.
No problem.
And your second question, Tony?
No, that was pretty much. No, my second things were questions were all to do with detail, specificity. There's recognition of a chain length that relates to the oacetylation of the N-acetyl mannose-amines, and I think that's an aspect to look at, if you like, in more detail. But I think that's a follow-on from what I was saying before.
Mm-hmm.
So what you've shown, Barbara, I mean, you picked this up because you were saying this is mainly structural, and what about the enzymes? But what Barbara's shown is how important the structural aspects are in all this, and how important, techniques we take for granted here, like SECMALS has played such an important role in all this. And one thing you mentioned, Barbara, was the fact that working in water, you saw this 20-fold increase in viscosity. Do you mean pure water with no electrolyte, just water with nothing else apart from the polysaccharide like conjugate? Is that right?
Yes, there will be some residual cations from the polysaccharides in the ampoules. We always try to make sure we know how they were manufactured, but if we add additional calcium, which is awful for freeze drying, the cake doesn't form very well. Sometimes we compare the stability of these freeze dried with a very dilute sodium chloride, but in the end, we always have ended up doing the freeze drying of this international standards in water, relying on the residual cation for some stability. And I think they're okay because the MENX is one we got from Finley Institute in Cuba. They were the first to make it. And at first, it was flying away off the spatula. We then actually, I said, we need to refreeze dry this with some sodium chloride. And after that, it behaved fine. We could actually measure it on a balance. And all of the polysaccharides that we have as those standards, you can measure them. But yes, in water, and it's not an intuitive thing for me to do, but some people say that they get a better recovery in water. But we used two different columns. We used a TSK column, which is our standby, because actually Connie got us onto that. The combination, 6,000, 5,000, and sometimes 4,000, depending. And we then used, because Kevin, as you know, pushes the Shodex columns.
Yeah, Kevin's the wire representative. but we were the first in, you know this, we were the first in Europe to get SECMALs alongside Walter Kuliker in Hamburg. But when we got it, we were not experts in chromatography. We were good light scatterers, but we kept on doing mistakes and putting huge back pressures through the poor little Wyatt cell because of the poor chromatography. And that's why Connie was brought in. Although we were lucky, we basically cradle snatched her from Chem Biotech in Birmingham, John Kennedy's, not John F. Kennedy, but John Kennedy, the polysaccharide man lab, and she transformed us with, as you point out, a knowledge of chromatography. But in just pure water, you're going to get large apparent viscosities anyway because of the primary viscosity effect, where you've got unshielded charges on your molecules. So that's where that comes from. But yeah, it's great how you show that physical measurements were very, very important, alongside the enzyme stuff, of course, Tony. Which
thank you, Steve.
But that would make Barbara's talk even more comprehensive, and yeah, we don't want that. We want the structural stuff.
Yes. Sorry.
Good. Any more quick, Gleb? Any question for you or anybody else?
Oh, yes, there's a question from TM. Oh, Thomas, you said, sorry, you don't have a microphone, but just had a quick question. Let me try to get that up. Oh, hang on. When you compared MW, weight average molecular weight, with antibody titer, was that a single antibody structure to one epitope, or an increase in the overall pool from lots of different antibodies to multiple epitopes within the larger structure? We looked at, I will send you the paper, Thomas, where we looked at the different isotypes, but it was an overall pool. First we did IgG, and then once we saw that, we then focused on IgM, IgG, and IgG1 and IgG2A, and I think maybe 2B or 3. So yes, it's a polyclonal epitope. Do you have a follow-up, Thomas? In the next part, which is just about 6 slides in the typhoid, I want to show you what we've done with comparing the human polyclonal with a monoclonal. You might be interested in that. It's not based on size, but it is an interesting thing with the O-acetyl. Thanks. Okay, great. Cheers. So now, now that, see.
Oh, do you
got to share this again. Okay.
Okay, you're to share the talk again.
Yeah.
Is it on the end of the same talk?
Yes, it's on the end of the same talk.
So you got the square with the arrow in the top right-hand corner. You got it?
Yeah, you see this?
Yes.
Okay.
Yeah,
there's just a few slides because it's now it's a new vaccine, and actually some work has to be done and better understanding of the behavior. It's terrible. What's happened with the VI, it's really a huge challenge. Putting it on a carrier protein is helping a lot, but they've got to get that from the fermenter onto the carrier protein. This just shows from 2014, some of the study sites in the blue dots and the endemic areas in the medium and dark red. And typhoid fever last year accounted for 9 million cases. It's a really debilitating thing. There's been the first pre-qualified vaccine for UNICEF's use, and it is being rolled out, but we need many more manufacturers, and dozens of manufacturers are attempting to make this with a paratyphy and with other O-antigen enterics. But this is the polysaccharide used in the vaccine. A vaccine was made in the late 1800s and used in the First World War, but it was very crude. Polysaccharide vaccine itself isn't great. It doesn't have great immunogenicity. But that's why it's called VI, because it's one of the first virulence factors. Interesting structure, it's N-acetylated and O-acetylated at carbon 3, but it binds to all the matrices, and it's really a pig to work with. There's the uronic acid carboxyl there. And the importance of the O-acetyl may be related to its position with respect to the saccharide ring. And in both Men-A, where it's essential, it's coming off of one of the main ring carbons, and also here. With Men-C, it's not. Sorry, here it is. It's coming off of this tail here. With the VI, here it is, the red, and it's coming off carbon 3. And it's absolutely essential. This is the only data that I'm showing you. It's a compilation of a lot of experiments using coating plates with either the VI of the native Salmonella typhi or Citrobacter freundii that have been deoacetylated through base treatment to make this panel. And it was then bound to human sera, which is an international standard. This was raised in Oxford for this purpose, or sheep polyclonal or a mouse monoclonal. And if you look at the 2 polyclonals, you'll see that at 52% and above, and even a bit below, there's still very good binding. And yet the cutoff for manufacturing is 52%. It could be lower. It may be better if it's lower. Other epitopes appear to be possible, and this has been confirmed since we published this last year by Duke University, who've done some biophysical measurements. The mouse monoclonal, clearly, it's like a monophasic. As you deoacetylate it, as you deoacetylate the VI, the mouse monoclonal isn't binding anymore. So it really demonstrates the immunodominance, and it goes down to 0. Now, this was done again in Cape Town, and it was done with 6 and 20 repeating units molecules. And here is with the O-acetyl group. It's a rigid helical conformation, and although you can't see it in this picture, if you look down on it, all you can see is the O-acetyl. If you take the O-acetyl away, it's much more flexible. These are the percent of time the conformers are expected to reside in that based on the time of the dynamics. So very important to keep the O-acetyl, but it could be a mechanism whereby other epitopes cannot be actually seen unless, even if you take it off, the carboxyl group and the N-acetyl aren't really that great, but once it begins to move, they then accessible. So that's really all I wanted to say about the conjugates. I've put on the back of this a list of a few recent publications from my institute on COVID, and this paper is really fantastic. Tracked coronavirus, which is shed in sewage, shed from people in sewage. And it was a way of looking around the world at the changes in how the virus, even after isolation and no travel restrictions, the virus is mutating in similar ways in different parts of the world. I'd recommend that paper. And these are all of the standards that are in the catalog for people that need them, usually clinical trial only, probably for these human antibodies. But there are also other diagnostics. This is a verification panel, so it will be sort of for the antibody assays that you can buy as a kit to give you high, medium, and low. So also different controls and nucleic acid reagents, RNA, and some research materials, which I just wanted to bring to your attention. They're only in small amounts. The spike and the nucleocapsid protein, which people respond to as well. And Also, I want to say that going back to the smallpox vaccine and thinking about what choices we will have. And yes, people have a choice of having the vaccine or not having the vaccine. But when somebody really desperately wants to prevent disease in their family, their community, and themselves, I think that they have to consider that safety isn't, it's a balance between safety and immunogenicity. And these notes on the left were in Jenner's hand, and what he said about the first boy, and he used a boy who, as you from the first slide, was more susceptible, and it may have been a progressive thing to use a child because their lives were not really valued as much as an adult who was a breadwinner or a caregiver. And he said on the 7th day, this James Phipps, he complained of uneasiness in the axilla. And on the 9th day, he became a little chilly. He lost his appetite and had a slight headache. And during the whole of this day, he was perceptibly indisposed, and he spent the night with some degree of restlessness. But on the following day, he was perfectly well. And after 6 weeks, he was challenged with smallpox, actually not cowpox, with variolous smallpox. And he was protected. And a few months later, his continual protection demonstrated persistence of immunity. I find this myself and all of the cases that they did, and they looked at, he used a very slight incision. They played around with the toxicity, the deadliness of it with the injection. And I think we can learn from going back in history. So I'll finish by just paying tribute to my colleagues and co-workers and my collaborators. Also, Steve, to you and your students and your group. And also to the director who hired me, Jeffrey Shield, who passed away a couple of years ago, who I credit with my career. So thank you very much.
Thanks, Barbara. That was fabulous.
Yeah.
And unmute now, folks. Unmute. Unmute, please.
Thank you. Thank you. You're very kind. Thank you.
It was fabulous. It really was. And yeah, Schild, I think, is when Tony does German. Schild is does it mean?
It means sign.
I thought it was a shell, like so Schildkreuter is like a tortoise.
That's right.
Oh.
Yeah.
Okay.
But it can mean shield.
Yeah. So he was
the same way as protection.
Yeah.
Yep.
He was your director then, Barbara.
He was my director, and he hired me and he supported me, but also all of the scientists, and he was really fantastic. He brought in a lot of projects for us. He was always enthusiastic. We had a lot of arguments. It's like that. And when his daughter came last year, I gave her a tour. They're Norwegian. His wife was Norwegian. He retired to Bergen. She teaches at a university in Sweden. But we remembered how in his cabinet, he had the whiskey bottle, and at all of the heads of divisions meeting, they all sat around with whiskey and a pad of paper, and that's the way they did things. And it was the old days. But he did come when we had a new flu building for flu reagents opened. And I did have a chance to thank him then, but I think sometimes he's looking on me now as well.
Well, our former colleague, when Tony was in with me in the Department of Medicine labs in Bristol, I mean, your boss was John Clapp, but mine was Michael Creeth, and we were all close together anyway. But he also had a whiskey bottle as well, but that was only used for times of emergency when things were perilous in the laboratory, so it was used for a different purpose. He used to keep it in his top drawer, little bottle of whiskey. You didn't know that, did you, Tony?
Yeah, I never saw the bottle.
Yeah,
No, I remember.
Maybe I should do that now.
Oh yes, I remember mine very, very well. Yes, yes.
I did locate my whiskey yesterday, but sadly I'm stuck here at work, so I can't pour one.
No, Vlad's our whiskey man, aren't you, Vlad? You're a whiskey man. It's good. It's in the tea room, I think.
Yes, it's not here. Yeah, I put it away from me.
That was fabulous, and it's good that the NIBSC have its supply of standard chemicals, standard materials, which I guess other laboratories can buy, or do they put a case forward, or what happens, Barbara?
If you are a national control laboratory, you get them for free. Other people may have to pay £80 or something like that for them. Collaborators normally get them free. If you want to make a case for a collaborator, that is at least how it has been. You can do that. So, but yeah, it is good because so the human serum standards, they will be encouraged to use for all of the clinical trials of the COVID vaccines. They will at least compare and bridge their internal standard with an international standard.
That's fab. So you're playing a vital role in all this this COVID.
Yeah.
So, and you quoted those 3-4 papers. Some names you recognised, Tony. Yeah.
Absolutely. I was just agreeing with you. Yes.
Yeah.
Very important.
Right, folks. I mean, we're hogging the discussion here, but has anyone got any questions for Barbara for the second part, or the whole thing she's presented? Gleb, you've been noticeably silent.
You're putting me on the spot.
Yes.
Yeah, no, this is absolutely fantastic talk and the information. I think I have a question. I mean, probably sort of not directly related to the kind of the overall spirit in terms of the confirmation, in terms of the sort of interaction between the polysaccharide and the protein fragments. So do we know about what interactions are sort of responsible for, and additional to the covalent links? Do we desire to have the polysaccharide and protein part non-interacting, so that essentially there is a kind of both of them have their own hydration shell? Or we would like them to interact by the hydrogen bond or the van der Waals forces in some way or the other? What is the desired conformation? So would you like to have a fluffy carbohydrate layer around it, or something more tight? Do we know any information about that?
No, but I would on it because, and I believe that it isn't, because there's something very interesting. And it is that it's not like a glycoprotein where things might be shielded by the polysaccharide. We've looked at this in a couple different ways. And we first, many years ago, when I was working on HIB-CRIM, actually, we did protease digest, and we ran them on SDS page shells, and we used non-specific and specific proteases like trypsin or a kind of pronase. And we looked at the pattern that we got from CRIM on its own and what we got from HIB-CRIM, and we really didn't see that the polysaccharide was preventing any proteolysis. Of course, those digestions were done at a slightly elevated temperature, probably 37, and that would have made it more mobile. In terms of something that wasn't done at 37, we looked at a whole panel of tetanus toxoid conjugates, all of them, a couple Hibs, a couple Men-Cs, W, Y, a pneumo, and a couple Men-As. So they were distinct in terms of manufacturing. And we looked at the effect of size you could say, you couldn't necessarily say chain length, but we looked at the effective size on the epitope accessibility by using 4 different maps. And actually, we did not see a correlation. In fact, there was a reverse correlation.
Right.
The larger chain length, the larger molecules, the larger conjugates actually bound the epitopes better. And I can only think that is because we did know from fluorescence that the protein was more open, but it wasn't like it was denatured or totally unfolded in any way. So based on that, I would say, and I would think that you want in the cell, you want with the processing for it to be able to be processed well and efficiently. So, and Steve, your paper didn't say there was a tight association. Not many people are actually commenting on this.
No, you mean the glycoconjugate? We showed that the properties of the conjugate were dictated by the polysaccharide component. That was based on the I think hide fit. We worked out the persistence length of the conjugate and showed it to be really as a polysaccharide rather than a protein. But that doesn't mean that the polysaccharide is tightly bound to the protein, stopping accessibility and things, which is what you're thinking about. It's just that the overall properties, and it doesn't mean that the protein was denatured. I think that was not at all. It means that the bulk properties was controlled by the polysaccharide. These were, huge molecules anyway, so it's not surprising.
Could I ask you?
That was the hip and the men. I think found it for both.
Okay, well that's interesting. I wanted to ask you about the technique because when we concluded with, I'll say, optical spectroscopy, which is strong for proteins and not for polysaccharides, where we found that some of the properties of the proteins carried through into the conjugates. I then thought that those techniques are biased for the protein. And although they do allow you to look at the protein once conjugated, they're biased. So conclusions have to be drawn with that in mind. And I wondered with sedimentation methods, are they stronger for proteins or stronger for polysaccharides?
No, but don't forget the hydrodynamics tells you something different. It really is about the overall properties, not so much what's going on inside. And if you've got largely polysaccharide, then it's not going to make much difference if the protein has lost secondary structure or not really. So I don't think there's any conflict in what you see with CD, with what you see with the hydrodynamics. It's the overall gross conformational properties with what you're measuring with hydrodynamics, especially when you're dealing with these complex types of conjugates. So I don't think there's any contradiction at all. What you're getting is this complementary companion information. The systems. But I'll need to look back at those papers because it was some years since we published that stuff. When Ali was a PhD student with us, so it must be about 5 or 6, at least 5 or 6 years ago since those papers appeared. That was just what we found anyway.
Thanks for that.
And of course, it depends on the loading as well of how much protein you load with the polysaccharide, not only on the take-up factor. Interesting. And it's also important what you said about the effective size as well on the immunogenicity of these complexes. So, and the distribution as well. That's why you have all these issues with the regulatory authorities, the patent bodies and things about the importance of not just the chemistry, the enzyme and all this sort of stuff, Tony, but also the physical properties have to be clearly definable. When you're dealing with molecules that don't like to be defined, and that's our polysaccharides.
Yeah, I mean, in a molecule like a tetanus toxoid, and what I believe was the case with the one that we had to investigate, and which we investigated at different points, is that in the smaller molecular weight peak, you can actually have a higher polysaccharide to protein ratio. It may sound counterintuitive, but if you bind it all up together, it may not conjugate more. And that can be a problem because there was a case that the FDA published on a long time ago. There was subpotent Hib vaccine, and it was because the conjugation points were too close together. Right. So they couldn't be processed. You didn't have a long enough epitope to actually bind to the MHC. And what could happen is if a manufacturer or just a researcher wants to improve their yield, they may throw in more polysaccharide. But you have to be careful. And in developing it, you need to go across the peaks and test the immunogenicity of each of the peaks and the characteristics.
Right.
It's just good practice.
Yes. Yeah.
Yeah.
We follow your guidance on that one, especially coming from the NIBSC. You can't.
I don't know. We're not. Well, I'm part of a regular. I mean, the MHRA are the regulators, but we're somewhat independent.
You're the BBC or the W.H. Smiths of the field in our BSc, so we have to conform with you guys.
Not so sure about that.
So, any more questions for Barbara? Because we've 1.5 hours. We've really given you a run for your money here. Not that we're paying you, but any other questions for Barbara? Quick ones before we go. No. Okay. So once more, let's unmute and offer our apology, our great appreciation.
Thank you. Thank you very much.
Thank you.
Nice to see you. Nice to meet you all.
So we'll just stop the record now. So we're off record, and then we can stop recording.